Hello everyone, myself Dr. Toral Vaishnani. I'm here today to give you a brief about basic informatics tool for NGS. But believe me, my topic is related to informatics. I'm not actually going to show you how a tool works and how we run each command. Because if I do that, you people fall asleep. You already have one reason to have sleep after lunch, and you will get second reason for doing that. Right? So it will be like I'll be giving you an overview of what is the basic how tool work and what are the most common tools which are used in the analysis process. Commercial tools are many, but I have not taken all of them into consideration. Not all of them. I can say I have not considered them. So we'll be talking about freely available tools, and I'll just name few of the commercial which are actually being used in the diagnostic industry. Your topic is again NGS, so it covers everything. But as we are from the diagnostic field, so this presentation concentrates more on the tools which are used in the field of diagnostics. So what I'm going to cover is just a general workflow, tools and databases, list of tools, variant annotation, and the hot topic variant interpretation. This slide already covered. This was the end of a session where he told us about the basic workflow which is followed for the analysis. So I know you mostly would be from the wet lab background or in the pathologist, right? So it's actually difficult for all of us to understand this flow. But as Dr. Neeraj already gave you a brief overview of that, we start with the DNA fragmentation, means this is the library preparation which happens. After that, it's the carried with the sequencing, and then you have reads in FASTA format. Now, this FASTA format up to this stage, it is known as primary analysis. But you from the sequencer again, you don't get directly FASTA format. In the sequencer, you get it in form of BCL file, which you need to convert it into FASTA format. So that one step that consists of the primary analysis, followed by the filtration of the data to remove adapter sequence and inequality sequence. Then, when this data FASTA is taken along with the reference genome, it can be HG19 or HG38, which we should all prefer because that is the latest version of the human genome, having correction made on HG19. So when this FASTA is used along with that, the format which you get is BAM file. That is known as BAM file, that is binary alignment mapping file. After that, when you use your tool for variant calling at this stage, you get VCF file. Now, what VCF file will contain? It will just give you idea which variants are called. That means it will tell you that at this particular position, it was A in the reference, but in your sample, it is T. Or half of the portion is A and half is T. That is heterozygous variant is called. But after that, what else? You cannot make out what that variant is going to do. That's why one more important step is annotation of that variant. Annotation in the sense, it gives you proper naming that how this variant falls into which gene. If it is falling in that particular gene, is it in a coding region? If yes, it is in a coding region. Is it affecting a protein or it's a silent mutation? If it is affecting a protein, then how it is going to, affect its function. So this you can know after annotation and interpretation. Interpretation is mainly when you have all the annotation with you, but now it's your responsibility to report only the one which is relevant to particular phenotype. Right? So that is done mainly in the part of interpretation. You have to interpret because all variants which are called, they are not pathogenic, they are not disease causing. So you have to narrow down it to report only the one which are relevant. So this is the basic overview. So now, why software and tools are required? When we go with the single data, you just need a simple program to analyze it. But for NGS, there are a list of tools, and you have hundreds of publications in a year. So why so? Because If you will read your DNA, it takes 9.5 years to write it in a book form. So if you want to read it fast, if you want to know what's in your sequence, so what happens? This NGS technology, it helps you break down your molecule and read it simultaneously. So you get so much amount of data or single piece. When we say 100x coverage, what do you mean by 100x coverage? That means you are trying to cover each and every part of what you have been enriched. It can be whole genome, it can be exome, but whatever is enriched, that is sequenced 100x times. Right? So that much amount of data is generated. So how to handle it? You cannot just be looking at the narratives. That you'll make that what this is going to make difference, right? So for that, for handling this type of data, you need a tool. So this is the case right now, and that's why we take a help of a tool. So as I told you, once we get the data from the sequencer, it is actually in a form of DCL. So what happens? Sequencer is having a capacity of like generating 4 TB, 8 TB of data, as Dr. Neeraj already said, right? That Novasic it's coming with the capacity of like 8 TB. So if that much amount of data has to be generated, number of samples goes together. So how they goes together? They have a different barcodes attached to it, right? This is like yellow, this is like one type of barcode, this is other type of barcode. So it's all mixed up when you get. So once you get that total complete run data, you have to demultiplex it to find out your single sample, separate your all samples. So this is the way this process is known as demultiplexing in bioinformatics. So multiplexing is when you are combining the data. But once you get the data, again, what you are trying to do, you are separating all of them. Whatever you have pulled, you are taking them back. You are separating them using the barcodes which have been added. So now, to understand the basic of all the tools, it's very much important to understand the format. It's like we all being very different person, we understand certain type of thing. We are able to speak certain type of language. So that type of limitation is also there with the tools. They understand different type of formats. They have certain limitations, or you can say they are more focused on certain part. That's why they have a certain format which they are able to understand. Okay? So this all are the formats which are taken by one of the other tool to give you meaningful variant interpretation. So now this we talked about, that FASTQ we get after multiplexing, demultiplexing, right? So you have a FASTQ file. Previously in most of the assembler, most of the sequencer, like Roche and all, you used to get FASTA file, but now we have FASTQ files. So once you have a FASTQ file with you, we filter it to remove the adapter and low quality sequence. But after that, we take the reference genome and map on it. And that is the basic step of all the analysis. As we have a reference, we map on it, right? So once you map it, you get the data in form of SAM file. So SAM file is nothing different from SAM file. SAM and SAM both are same. But it's like one is the binary version of SAM file. If you want to open the SAM file and read it, you can do it. But when you convert it into binary format and reduce the size of the file, after that, you will not be able to visualize it directly. You have to take a help of tools, like samples, a view program of samples to visualize that. Then once you have done, you use variant coder on it. As I said, there are no pattern vectors, right? If your interest is germline mutation, you need to use tool accordingly. If your interest is somatic mutation, you have to use tool accordingly. They work differently because deployed thinks that it's you're working on germline works considering that you have deployed genome, but somatic works on the different principle. So that's why variant coder differ, and then you have a VCF file. Now, once you have a VCF file, again, you have a conversion from T to C, whatever conversion is that. But then to know that that variant belongs to which gene, for that, you need the help of GFF file. Now, GTF and GFF file is nothing, but that comes along with your reference genome. For example, if you're using FG38, then that, along with that, you have a file GFF or GTF, which have information of all the genes. It is basically, you can say, address of all the genes. It gives you information about all the genes, that this gene belongs to which chromosome, which position, where the exons are located, where the introns are located. So it's basically address of all the genes which are present in the particular genome. So just a brief overview. FASTA you all know, right? It starts with the greater than sign and it is followed by the sequence. So this is the thing which you get after single sequencing, or as I said, previous sequencer used to give data in this form. So this is a very basic form of input data which we give to the any tool. Now this is FASTQ, as a name suggests, FASTA plus quality. That makes FASTA to FASTQ. So it's like a line, you have a header, followed by you have a second line of sequencer. In FASTA, you have two lines. First is header, followed by sequence. Now here you have four lines. Why four lines? Because you have quality into it. That's why you have sequencer header, then sequence, then it's just a separator, and then you have quality for each basis which are shown in the sequence. So let's see example for this. And I think this is very basic thing we all know. When we talk about forward sequencing, it is like R1, forward sequencing, R2, which is reverse sequencing. That's what for all sample, if you consider single sample, you have two files for each sample, bioinformatics analysis, right? So each sample, two files, forward and reverse reads. So this is the example FASTQ file. This is the sequence ID. Basically, this is like your instrument name, ID, then flow cell, which flow cell you have used, and then this is the barcode information which has been added. So demultiplexing tool like BCL to FASTQ, which is of Illumina, they separate your sequence based on this barcode sequence, right? Then this is the normal fragment, which is red, right? Then this is the connector, and then this is the quality. Now, quality, though it seems like it is alphanumerical, but it actually represents number 1 to 40. Right? If you see Fred encoding, you'll see that this quality represents 1 to 40 number, though it is alphabet, but it represents, and special symbols, but it represents 1 to 40. In FASTA, this has been converted like this just to reduce the size of the file, nothing else. Right? So here the three symbols which have been used, they are explained like this symbol represents quality of 2, which means it's not good. So you need to trim bases like this, which are of poor quality. Then this A, which is represented with 32 quality, which is actually good. If your data is Q30, more than 80% of your data is Q30, then you can say your data is good enough to start with the analysis. Right? So this is just a outline of the one FASTQ file, but we have the tools help us in analyzing all these stats with a complete file which is having millions of data. Means millions of reads are there in the FASTQ file. So that tools helps you get all this information from your file. Now we have FASTQ file, but how will you know? As I told, if there is a quality like 2, you cannot see if millions of reads are there. Can you see manually? Iski haan read ka 2 hai, iski base ka 3 hai. We cannot go like this, right? There has to be some way. So there are many tools which are there for quality checking. This is one of them. The tool name is FastQC. It helps you in analyzing the quality of FASTQ files. So this, as you can see, which is good in quality? Which I will mention in the figure. But if you can see, this is, I don't know whether it's British clear or not, but this is the quality value, 0 to 30. Right? So you can see this bar is upside here, where this here it is coming like this. So this shows that many of the bases are just of the quality 2, this is 4, this is like 6, 8. So this is poor quality data. And this is the graph for complete FASTQ file. So from the whole file, you are getting this graph and you can understand whether the data is of good quality or not. Okay. Then we have a FASTQ file. Now we have a reference. Then I tell you that from the FASTQ, from the reference, you get BORN file. But how you get it? So this is just a graphical representation, as I cannot show you that Linux BORN format because you will not be able to understand that. So this is just a graphical representation of that once you have a sequence here, and this is the human genome which you have taken, you can consider this black as a human genome, and these are the reads which are not bound to it. So it's like you have a reads in form of a puzzle, and you have a reference genome like this. Using this, you are trying to reconstruct this. Right? So when you talk in terms of sequences, you'll get something like this, that this is the reference genome. These are the reads. One read, two read, second, third, fourth, fifth, like this. But as you can see here, in reference, it was T. But here, in the particular sample, it is C. In one of the sequences, it is T. And again, it is C. So it is a mutation. Heterozygous or homozygous, that again depends on the reads. When you analyze, you have to, after that, you get to know about the variant allele frequency. If half of the reads is representing the reference genome, that is T. If half of the reads are representing T and half of them is representing C, then it is heterozygous mutation. If all the reads, here it is just 10 reads, otherwise you can consider it like 100 reads. If all the 100 reads are representing C here, and in reference it is T, that means it is a homozygous mutation, right? Now, this is the actual SAM and BAM file. There are, for example, if we have 1 million reads, then in SAM and BAM you will have 1 million lines. So this is like the read ID, the read which fast to read, which I showed you, that is the header, right? So here it is a header. It tells you that how it has mapped. Then it is on which chromosome the read has mapped on the particular reference genome. At which position, if it is chromosome 10, but which position? Then we come to know from there. Okay, if it is mapped, but whether it is able to map confidently. So if quality is 60, like 60 is like good quality. It's mapped with a good confidence. So this is mapped in a proper way. But if it is 0, then you can say that it is multi-mapping. Multi-mapping means it's not confidence. It is mapping to this particular part of the reference genome. It is also mapping to other part of the reference genome. So that is less confident. Right? So this is basic overview of the BAM file. Now, GTF format I told you that it is actually gives you a basic address of the gene. I'm not talking about the variant. This file comes along with the reference genome and it is having an information about the gene. Structure like exon, intron, UTR, everything, right? So you can undertake it, consider it like this, that it will show that okay, this is the chromosome. So it will have a position like chromosome, starts with this. And here it will end from the region line, then this exon will start. So it will give coordinate information. So basically it will, GTF will have coordinates for the components of the particular gene and genome. Now the VCF format. The thing which I showed this previously also, this is like a graphical representation. But when you want to see it in a form like 20,000 sequencing, you end up like 30,000-40,000 variants, right? So it is in a form of a file where they are listed one by one. So how they are listed? It's like this. So this is the VCF format. It starts with the chromosome number, then its position. Its RSID is present. It depends on the tool which you are using. If you are using a tool in a way, you are giving its DB-SNP database in the bucket. DB-SNP database, I think you must be aware, right? So if you are giving that in the database, then what it do when it calls the variant is check that position with the DB-SNP database which you are given. And if it matches, then it assign the RSID here. Okay, if you are giving cosmic, it will give you cosmic ID here. So whatever database you are using, whatever your interest is, based on it will give you your information. Then it is what is in the reference, what is in the alternate, right? So here, when you say G2 is genotype, so when you say 0-1, it's like heterozygous. How heterozygous? Then it will have reference and that means that variant, which is called, is having support of how many reads? 213 reads. From that, half of the reads, almost half, it's like 105 reads are supporting the reference genome base, and 1 is representing the alternate base, that is A. And that's why it is heterozygous. But here it will see, it's 1/1. That means it is homozygous. That's not this homozygous, where you have 0 reads representing this 0, it's representing the reference allele, so it's 0. And variant is like alternate allele is 113. And that's why your total is also 113. So this, from the VCF itself, you can come to know whether your mutation is heterozygous or homozygous and what is the frequency. Frequency is nothing but it's like division of this alternate allele into 100 divided by total. So if it is germline, it's like 40 to 60% basis you get in most of the time. It is homozygous germline, so that's why it's 100%. So your work is variant allele frequency is 100%. Now the list of the tools and databases. So just to begin with, as I told you, first when you get the data from any sequencer, first step, irrespective of any application which you are using, it can be diagnostic, it can be research, anything. But first step is demultiplexing. Second is you check the quality of the reads using the fast QC, the graph bar chart which I showed, box plot, right? After using that fast QC, you take a decision that how much trimming is required. So you trim the data. That's why once you have trimmed it, you'll map it onto the reference genome. So these are the aligner which helps you doing that. BLAST, you all are aware of the BLAST? That is also one type of alignment. That is alignment, right? But when you have that big genome, this mapping comes into play and it counts all the million number of traits to go onto the reference genome and give you an idea of how it is, representing that particular genome, which part of genome it is representing. So these are the aligner which commonly used. Then this mapping duplication. We know when we do the wetlet procedure, then you get also PCR duplicates. So when PCR duplicates are very high in number, that example we took 213 sequences were representing variant, right? From that 213, 50% is PCR duplicate. What you feel, PCR duplicates. So what you feel that variant has been covered very nicely. 213 reads are representing the variants. But the cases, the other 7 reads, they don't add anything to that, right? So that's why you need to first remove the PCR duplicates. So tools very advanced there. So based on remove the PCR duplicates from that, and then you get the new file, which is again processed, and that is used for direct column. Now this is sequencing matrix. It's like after you get the one file. Now one file is also big because when you have millions of reads, that one is a representation of million of reads along with the reference genome. So it's very good. So now how do you check the quality? Fast Q sequence, you use fast QC tool, right? So here is a very number of tools which gives you an overview in a beautiful graphical way from the insights which you consider while sequencing, how reads are mapped. So all the metrics you get from here, right? Using this type of tools. Then you have when whether you want to join. So the main terms will this works on a different basis, right? So these are the terms which are used for that. This slips and enders. Now we know that copy also plays very much important in the disease. So that's why the delicate tools for that. So few others from the exome CMV some of the widely used. Then you need to check the structural variants. It's like translocation of everything you have to see. These are the terms which gives you that this three majorly used. Then which is in trend, fusion study, which is very much important for some of the disease like ALL. And then the gene fusion analysis, which is based on the RNA and DNA. So this all things works on the DNA, but it works on the RNA. That's why RNA fusion mostly studied using RNA-seq data. Then this is visualization and review. Visualization, non-competition, or you can say even competition. We cannot look into each and every region and variant how it has been offered also. So that's why we have to view it in a way that we understand. So for that, there are tools which helps you view the variant. I'll give you example how if you try to view it, how it looks like. And for this BC, once you get the VCF and BCF manipulation which can be done. For example, if you want to remove any variants which are, which are having variant having frequency less than 5%, because you know that the color, the sensitivity of all the that assay is more than 5%, right? It cannot pick up the variant which is less than 5%. So in that case, if you want to filter out that, so that you don't have to look at that junk. So this type of tools helps you in its manipulation of the VCF file. Right, so, and this is for annotation. This is really important, as I said. If you get the gene address from the VTF file, but how do you get the variant address? You get that only within the annotation, right? So these are the tools which are helped in annotation. VEP is widely used. WNP you can use, that is the best tool. For example, if you have got the VCF file from any company and you want to check the variant of your interest, but you actually don't have complete data with you. You just have a report sent by them. So if you want to do it by yourself, you can use Bioinformatics program and VEP. You can upload your VCF file there and can check out the result. So how to use these tools? Mostly they are Linux based. With this knowledge, which is actually very typical implementation. When you look at the screen of bioinformatics person and if you sit beside them, you'll feel it's very boring. But for bioinformatics, it is actually very interesting. When you see the left, it tells you that this is done, and any particular tool is, doing analysis, it will give you this part is done, this is done. So when you see from the distance, being a non-competition, sometimes you find it's very boring. Sometimes you feel it's a rocket science, something is happening. Like maybe you see that cycle, maybe this clicking and it's giving a result. That thing happens in bioinformatics. But for non-competition, this is very much a good platform. This is Galaxy platform. So if you have a raw data, like in a form of a FASTQ file, and if you want to do the analysis by yourself, this is the tool which can help you. Here, I have just highlighted this mapping and event calling, but when you open this, you have a number of tools which can be used for mapping. So you have to choose based on your need which mapping tool you want to use. The list which I have given is present here. In this previous slide, the list which I have given for mapping is also present here. So you can choose from here. And then green column. If you are either to use scientific column different, German column different. So you can use your list of columns there, you can select and do the analysis. So you will get the VCF file from there. After VCF, you can also interpret it using W. But you can use W annotation. So now CNV analysis. Very important thing. Now, how do you know this CNV is there? Right? So how do you work on what this is? So this is the simple scenario of this, that this is the reference to now. These are the reach magnets. Something like that, reach magnet. But in the patient sample, for example, structure is something like this. But we don't know what structure is something like this. You forget this. We don't know actually this. But this is just giving for the graphical representation to give you more idea. The particular patient has been replicated twice, right? But actually, we don't know that has happened. So how do you do? When you reach and see this. This is the control sample. Which are one chakra. Here you can say kinds of that is mapping. Right? So what does that mean? That and ref. Actually, when you sequence that sample, patient sample was having 3 copies. And that's why data generated for that particular sample is thrice. है ना? आप एक अगर कोई पेपर लेते हो that is of red color, एक कलर का है, तो आप उसको तोड़ोगे तो उसको रेड कलर की पीसेस ही you'll get. But if you take paper which is multicolor, उसको आप तोड़ोगे तो you'll get multicolor pieces. तो वैसे ही है, if you are taking control sample, उसमें सब कुछ एक जैसा तो you'll get तो समझो कि यहाँ पे सभी जगह पे क्योंकि you generated 100 data to complete enzyme. ऐसा तो नहीं है, it is a proof-based enrichment, so definitely there is less and high depth means you have some places where it is covered and some are actually less covered. So it happens in exam. That is the limitation of exam. But जब आप उसको सबके साथ कंपेयर करते हो क्योंकि कंट्रोल भी उसी से एनरिच होके आया है उसी ऐसे से एनरिच होके आया है still it is having same depth for the region but here you are having twice the depth that gives you idea that this is duplication अगर यहाँ तक कुछ नहीं होता it would have been deletion got it so duplication you get more reads compared to the normal and in the deletion you will get no reads no chance then it will be limiting 2 to 3 reads but not more than that okay so then it will be a deletion now fusion now how fusions are identified so this is just an example that if you have chromosome inverse and this is the B chromosome this second gene is inverse but now what happens when we have leakage this is the B part of this particular gene so this portion is being fused with this portion, right? So that's why this is the big point. And the roots which are on this portion, because you have one cell, you know where the roots are going. So if read usually eliminates of just 150 base pair, right? So if we take an ideal scenario, if 75 of base pair read is mapping to this portion and 75 roots are mapping to this portion, then it shows you that fusion has occurred. But it should not be just evident for 1 or 2 reads. It has to be more reads to show this evidence. And this read is mainly split read, right? Because this read has been split. It can be in one ratio, 25% on this, 75% on this, or it can be 40% on this, 50% on this. It can be anything, but the read had split and given you idea that something has gone wrong here, right? And there are other things. Spanning reads. Spanning reads means you have taken actually when you're preparing a library, you have taken this fragment, you are sequencing it from here, that is R1, you're sequencing it from here, that is R2. But a fusion has occurred, and this is your transcript because here you are taking R1 data, right? So if this is the transcript you are taking, then what will happen? This is fusion, half this, half this. So you have one read here, R1. R2 is coming from other gene. So when gene reads show this type of trend, usually it happens that your both read should go to same gene, right? In ideal case. But if your reads are showing this trend, R1 is mapping to this portion, R2 is mapping to other, that gives you evidence that fusion has occurred. So that's why with the help of this split read and spanning read, we identify the fusion. So this is the thing which has happened, right? So this have merged with this. Now, once you have a variant cell, every 40,000 mutation can be there in the exome data. If you talk about the genome, there can be 3 like 4 like SNPs. And how do you go through it? How do you reach? How do you to show that this is the useful thing? Right? So for that, you annotate the variant. Annotate is like giving address to the particular variant. So you have this genome. You have this chromosome, so you are tagging the variant. It is coming from this chromosome, it is coming from this gene. This is the seed that changes which is happening, and this is the change in criteria. So this is what you get after annotated this year. Now again, we have the annotation. But after that, what? Whatever variants are discovered, are they pathogenic? Are they disease causing? No, right? So how do you do that? So again, many tools are there which comes to your rescue, and many criteria which helps you guide that. So this is actually journal guidelines for journal analysis and interpretation. So if you can see ACMG guidelines 2015. So if you are given this 28 criteria, which helps you in the interpretation of the variants, whether they are pathogenic or not. From that, this is my part because it comes from the database. This comes from functional studies, from publication published, from clinician, from the history, right? So it comes from different places, but this comes from the databases, from the computer. So you can work on this aspect easily. So let's see, based on this, we are classified as pathogenic, likely pathogenic, VS likely benign, no. But there is a combination which has been considered. Like if you people come to know that this also criteria, TV shows very strong criteria. Very strong. Pathogenic supporting. So this type of this is like benign supporting, standalone. So these are the criteria. So you need to go into much detail into it if you want to understand it. So this is just an overview. P indicates pathogenic parameter, B indicates benign parameter. So one of the easiest way, if you want to reduce your data from 40 to 1,000 to 50,000 variants, one of the easiest ways to compare it against the population database. Neeraj sir also showed in the slide, they showed population frequency database which can be used. So that database is having a variant called from the normal healthy population. So what indication it gives you? That variance, because we are indication. Are we having some disease? Excessive mutation which we carry, but if you want to say that you are healthy, so gives us confidence that this is benign. So that's why we compare other variant permutation with the population frequency database. If the population frequency database is having more than 5% of that, means for example, 60,000 which is sequenced, right? So from that, 5% of the population is having variant which is also present in the sample. More than 5% is having this variant in this population, which is present in our sample, then that is eligible for standalone benign. That means that is polymorphism. That cannot be disease causing, right? So these are different diseases which can be used. If population frequency for this particular variant is very high in this, we can ignore the variant. Then comes this second criteria. Sorry, this is the
for polymorphism, I guess it is 1%.
1%. It's actually better to consider, means if you consider up to 1%, it's like supporting benign. If it is more than 2%, as I said, it is standalone benign. You do not need to see any criteria for classifying it as a benign. But for 1%, it's like almost directly benign, but still you can check other parameters. And if it is in points like 0.005 something, that can also, because it is rare, so it can be disease causing. So you need to check other parameters as well, other criteria as well. Yes, very much important. Yes, yes. So that's why when you actually, I thought it would be confident. That's why I did not try to do that. If you see, so there are different consisting partition, right? So we have South Asian. SAS is there. So if you are checking, so it's necessary to first check the global frequency. Along with that, you need to check for SAS, South Asian population, because some of the population is very much Indians. So that's why you can make decisions on that basis. So these are the list of the databases which can be used for that purpose for filtration of the variants. Then there are tools which helps you, because you don't have any functional evidence that this variant is actually causing change in the protein structure and that's what is causing disease. You don't have, functional information for every variant you do. That's why there are specialized tools which helps you analyze it in silico. It does some applies and it helps you analyze and it gives you, idea in terms of whether it is imaging, standard imaging, and tolerable. Right? So tolerable means the medication is not causing much damage. Right? So that's the best chances that it can be done. But it is a very probability. So you cannot just consider this criteria. It has to be some combined with the criteria because this is all in silico. It's just a prediction. It's not something which is observed. It is just a prediction, and prediction might be wrong. Whether prediction कैसे wrong होता है, वैसे ये भी wrong होता है, wrong होता है. Because when you take जैसे in silico prediction tool you have used, some of them will show it's benign, it's not disease causal, and some will show it as deleterious. Right, so in that case, you have to take decision. You have to consider it as it. But usually it is you should minimum use 3 in silico prediction tools, and based on tools, you should go with that decision. If 2 tools are showing pathogenic from the 3, then you should consider it as a deleterious. Yeah. No. No, no, no. Only just coils. It will do nothing else. So, yes, in that way, yes, it is definitely that is also, yes, because all the tools have very limitation. Especially if you see with the assembly, it's not a problem. But when you have deletion and insertion, most of the columns you the transport. So when you take a combination, when you call it using 2 to 3 columns, and when you use it to take only the consensus one that is present represented in 3, then there is advantage. That you can get. There is also disadvantage that you might miss the one which is actually true called by one of the column, right? So it's again one call. So basically, this whole analysis of everything is like it's nothing nothing like black and white. It's all gray. So every time you decision, and everything depends on your decision, and which experience matters. So that when I told you should take into consider 3 tools. One is the prediction. So like this are the ones we use this actually works on just net. This is the insertion and deletion. But this is the example shift. Shift this different just scoring. Shift gives 0 deleterious, and polygon gives 1 score when it is deleterious. Right? But the aim is same. But when different direction, it's like when it's 0, it's pathogenic. When it's both 1 score 1, it is pathogenic. So you have to remember which is pathogenic. Right? And there is also a score which which considers different parameters into play, and then there's one score. So if it is more than 15, it is probably pathogenic. So first population database for patient data, second is in silico prediction. Now third is very annotated disease database. हमें patient clean में मिल जाता है कितना अच्छा लगता है. We don't decide much more. We think much more. We don't decide सच्ची है या नहीं है. Because listed as a pathogenic. So you don't have to do much. Definitely you have to cross check it. You cannot blindly trust anyone. और जब 10 मिनट उसको बताया है जब जैसे हमने समय तक में जब तो 617 पोजीशन अब वो तो सबको पता ही है वो तो टेंशन ही नहीं है कि क्या है कुछ सोचो मैट्रिक्स पैथोजेनिक राइट सो so you can use criteria like that when that particular mutation is present in the clean database. There are number of databases, but one of them is clean one. In clean one that is present, जो 20 criteria थे उसमें से एक जो pathogenic वाला था, this is like PP5. This is supporting. That means you have got support from other people also telling them that it is pathogenic. So this adds into pathogenic criteria. So this gives you more confidence predictor is pathogenic can be is actually pathogenic. And when this is benign, so this criteria is added into the other criteria which you have considered the population frequency and in silico prediction. So this adds more into it. This is the third criteria which gets added into it. Right, now this is the difference. See, this is mostly general. This is general. This is paid. Paid database only. The stream and then again this is sensitive. This is also the concentration. This is basic or CNV. Now, this is the fourth criteria, which again depends on the tools and databases. This is the last criteria which you can define based on the using different tools and databases. I give you functional and other points. So this, this is normal. Now this function has changed to it is silent because the routine is changing. Right? It's prolonged to prolonged. That's why it is silent. Now this let's see TCC. TCC. So change to silent. So this is sense that you can say non-synonymous. Now this is friendship because this this shifted. That's why this is non-friendship. And here becoming this but results in that, and that's why that protein stops there. So based on that, these are criteria which can be applied. So synonymous and so you don't give pathogenic criteria if you have this particular type of variants. Right, so that adds to the point of pathogenicity. That increase 1 point to the pathogenicity. The shift can start again. Start again in the sense, your, for example, protein has 1,000 amino acid, but you got start gain at 300 down. Then now your protein is just of 300 amino acid compared to 1,000 amino acid in the protein. So now 1,000 amino acid protein works in a different way. How would you think 300 amino acid protein will work in the same way? Don't think so. It will work in the same way. So there are very so this is the just for some population frequency, this are the type of prediction, this is region, hotspot. If already mutation has been called from that particular region, then it is hotspot region. So this has been given. This is from received if it is prevalent in GMB and this is computational term. If all the terms, most of the terms which are then you get this criteria, right? Now you will know whether it's pathogenic or not. Chalo, आपने decision लिया. That this is matching with your phenotype. Phenotype is very important. When you competition, don't just talking about variance, but what is much important is phenotype. And as in the previous session, the doctor said, if you will not have phenotype, you will not be able to so it's very important to understand that maximum phenotype we give it to us, maximum information we give it right? So once you find that this is pathogenic from bioinformatics point of view, you need to check the quality of the variant. So how you will do this? I told you previously also which helps you in seeing the variant. So this is the way variant looks like. This is the reference. This is the reference sequence. This is your reads. This represents the reads. This is forward reads. Right? So these are the reads. And I can say, see here it is G notation, and here in reference it is A. So A is changing to G. And you can say total reads. Total reads, this is representing 1073. And you can say 600 and reads and G. That is this hypertext notation. G is being represented by 3 bases, right? So this is missense notation. It begins like this. Now you have insertion. Insertion, you get it in this form. This is the simple basis that gives you insertion. See here. Here you can see the number of insertion. So this one is the reads. This one is the G, which is reference and number of times. This is the reads representing insertion. Now, deletion. Reference, here you can see that here it's been deleted. Again, it's 1.30, deletion representing 830. Reads, right? So this is deletion, how it looks like in IGV. So now, when you have predicted that with the pathogenic, it is matching with the phenotype, and this also seems good. If the quality is bad, here also you will see mutation, you will see mutation, insertion, deletion. That means it is a poor quality. These reads are poor quality, and you can press that here. But here, as you can see, these reads are so clean. Okay? So that's why this is in quality terms also, it is good to go with. So you can report this variant now. So now to summarize. You have got the sequence, it goes into the sample because for handling this big data, I showed you Galaxy, right? But Galaxy will not be able to use when you take 10 samples, 100 samples, or 1,000 samples. So you need your in-house server, a big server. Because when you're talking about engineering, you are generating minimum like 100 GB sample. When you're talking about whole exam, you are generating approximately 10 GB exam. So you have to have storage capacity along with the processing capacity. That's why data directly goes into the server, which is having a good capacity. There it is analyzed, quality is checked, this is the factor, this is the quality check, then quality happens, prediction times, they have preparation of the times, they have received and its quality, it is then reported based on the phenotype match. Right? So this is the overview of what is being done by different tools and databases. Now, few questions, just a normal, it's like when you get a report from any of the company and it's negative. So first thing that you need to check whether the gene which you have given and is covered by the gene. Is that gene a part of the data? If yes, then how sensitive is that assay? Is it that sensitive to capture the long deletion insertion, which are main problem causing in that particular disease? So that you need to question yourself and decide, right? Then you do that. Then it is actually when gene excited disease movement, it's actually case in the scientific mutation, right? So however, that is affected many factors including branch of assay, sampling changes in concentration process and right? Also, just generally we expect the variant allele frequency should be 50 and 100. But sometimes it happens that you generally you get at 25% allele frequency. So if it is matching with the phenotype, based on your experience, you should report it. Or you can talk to the clinician, you can talk to the family, you should then confirm it in the family. Segregation analysis should be done. Right? So based on the decision can be taken. Then different symptoms and so this is just a basic normal culture which is used for representing particular event, right? So this is a coding position which has been shown here. This is the this is also found. And this two documents, it's a protein change. It is talking about that this we have 600 codon is going to change from V to E. Here it is shown to be codon. Actually, there is mutant which is called altering the region, and that's why this protein is getting altered. Right? And if you have a fusion, it can be shown like this, and even LB. So there are certain ways of representing a fusion. So if I'm familiar with the variant and the report, many of you must be receiving a report from the company. So if you're not able to understand what we mean to say, you can check it yourself in number of the databases. This are mostly somatic based databases, right? So you can check your notation, whichever has been reported done here. And still, if you face problem, call them, ask them. Okay, and if you feel something is missing, ask them whether they have covered this particular genes, whether they look for those genes, because see, when clinician gives a phenotype, the set of gene, because if we take it in genes, many genes get covered. Then set of genes which are of interest, that's not allowed. So once you receive a report, all the genetic also again ask your therapy to, if in case if you can give answers or check for the particular gene, they have checked it or not. If they have checked the mutation, right? So that's done. So this is just a reference. Thank you. Any questions?
No, I just want to, before anybody asks any questions, it has been a brilliant presentation. I must compliment you.
Thank you, sir. And should I give some credit to this course,
Oh, yes.
GYC course, which has been carried out by Dr. Vinod Sakaria? So some part of it's taken from there. And with the guidance of Dr. Neeraj Arora, this has been prepared. So thank you.
Good.